Good morning, ladies and gentlemen. A very warm greetings to each and every one of you. My name is Lindsay. It's a wonderful and precious change for me as MC for this event. So, ladies and gentlemen, we welcome you to the first session of Workplace Proposal 2022 with the topic the impact that can improve academic immunity. As we know, cancer causes high fatality rate and is ranked second for the cause of death in the world. Before we start our event, let us start by singing our national anthem, Indonesia Raya. For the audience, please stand up. Okay, the audience can sit down now. Our event today, Workplace Proposal 2022, is held by Chemical Engineering Department of ITS. So I welcome Mrs. Widya Stuti, P.hD, as the head of Chemical Engineering Department. I welcome Professor William Michael Cheng, P.hD, as our honorable guest speaker from National Cheng University, Taiwan. And I welcome Mr. Hakon Wirahastito, P.hD, as our moderator today. So without further ado, let's welcome our head of Chemical Engineering Department for deliver her opening remarks. Mrs. Willy, please welcome. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Welcome to ITS, Prof. Chung, and also, I'm sorry, Michelle. Okay, nice to meet you. It is honorable for me to welcome you to join this lecture guest. It is very interesting that the topic is the compound that can increase under cancer immunity because we have many uh, source that can implement it for cancer disease. So I know that Professor Chan is the expert in the cancer, also how to get to overcome this disease. So it is a pleasure for us to invite you as a guest lecturer, and also Pak Hakun, thank you very much to accommodate that this event from this is a part of world class professor. So I hope this event is useful for our students, and also we can get to increase more intensive for collaboration. Maybe not only Dr. Hakun and also Prof. Gunawan, also other lecturers can also join for this event. I think enough for my speech. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Ms. Widi, for that energetic opening remarks. I would like to request Ms. Widi to present our department nominee to Professor Cheng. Please welcome. Still waiting. Thank you very much.
1 minute.
Okay.
So on the turn, I also prepared some gifts. So this is a very nice cup underneath,
Yeah.
and then this is a very nice chopstick,
Okay.
and huge bag,
Yeah.
and also some bag.
Thank you very much. Thank you very much.
Thank you.
Yes, thank you very much, Prof. Cheng, for the unexpected gift for us.
I should.
So before we move on to our main session, let me introduce our moderator. Here, Mr. Hakon Wirawasito, P.hD, will serve as moderator. Mr. Hakon is one of the lecturers in chemical engineering, focused in fluids and lipids, which is linked in some way with the topic for today's event. For Mr. Hakon, the floor is yours.
Assalamualaikum. Assalamualaikum warahmatullahi wabarakatuh. For the Dr. Widyasiti, for the Chairman of Chemical Engineering, our speaker, Prof. Cheng from Chung University.
Aku juga dapat di grup asal share aja.
And the ladies and gentlemen, for the speaker, Prof. Cheng speaker. We want to introduce our speaker. Prof. Chung with Young Picheng. This is from the Cheng University as a good speaker. The degree is the P.hD from the Chinese University of Hong Kong. The master in the Chung University, Taiwan, and the bachelor in the Chung University, Taiwan. The working experience, this is now is a director of international office in Cheng University, and for the working is a reviewer in the Kiwan journal. It's for our presentation and invited talk is in the Transitional Pension Economic Gordon Research Conference in Hong Kong, and in the annual meeting, the Japanese Pension Association, etc. For the time is yours, Professor.
First of all, thank you very much for the kind invitation from Professor P.hD and also Professor Chung that I can visit ITS again. So this is my second time visiting ITS. So this is this shirt was purchased last time when I visited ITS. So I'm wearing this shirt again, which is actually 2019 November. Yeah, so the COVID pandemic actually hurts a lot, but fortunately I can visit you again. So what I'm going to share, okay, may I have my PPT slide? Can I control by myself, or do you have any remote control? Unfortunately, we don't have any. Oh, I have. I prepared. Hold on a second. Check. Okay, it works very well. Okay, so okay, yeah. So so the talk today is I'll change a little bit. So targeting the epigenetic machinery to enhance cancer immunotherapy. So can I sit or I can stand? Or... Oh, okay. But is it good? Do I need to? Yeah. Oh, okay. I think it's better for me to stand around. It's more convenient to do so. Okay, so I changed the topic a little bit. I will try to speak slow because probably you're not very familiar with these topics. So basically, I would like to introduce some compounds that can increase our anti-cancer immunity, but through the epigenetic machinery, which I will introduce later. Okay, so firstly, for those who are not familiar with National Chung University, so we are located in Jiayi County, which is right here. You can see, which is 250 kilometers south of the Taipei city and 100 kilometers north from the Kaohsiung. So we have the high-speed Shinkansen bullet train. So it takes only 1.5 hours from Taipei and 30 minutes from Kaohsiung. So if you visit Chung University, you will see a lot of pineapple field. And I know lots of Indonesian students probably watched this Meteor Garden before. So most of the scenery was taken at National Chung University. So if you visit CCU, which we usually call CCU, So I will bring you to visit all those scenery that was taken this Taiwanese drama. Okay, so for those, although there are only a few that are attending in person here, but I have some gift to prepare for you. Those who ask me questions, so I will give you either the blue one or the red one. So it depends on who will ask me question. Okay, so you have to pay attention. But I'm sorry for those who are online, you cannot get my gift. But maybe I can leave some for the department, so that if you post your question on, you can also, if you post your question online, so you will also get a gift. Okay, it's fair enough, right? So okay, so the first two who post a question online will get a gift. But for those unlimited, as far as I have enough, okay? Okay, so let's go on to the talk. Let me introduce the tumor microenvironment. So this is the tumor cell. This is tumor cell. So actually, maybe 10 or 15 years ago, we are thinking only a simplified model of the tumor. But right now, we have more a comprehensive understanding of what a tumor is. So tumors not only contains the tumor cells, but it also contains, very important, blood vessel. So you have blood vessel to supply the tumor cells. And we have a lot of supporting cells so that the tumors can grow. For example, we have cancer-associated fibroblasts. But most importantly, what I'm going to introduce is the lymphocyte that infiltrate inside this tumor. So the lymphocyte contains T cells, B cells, or NK cells, which I will introduce a little bit later. That those immune cells play very important roles, either supporting the growth of the tumor cell or to restrict the growth of the tumor cell. So what I'm going to introduce is how those lymphocytes affect the growth of the tumors. But if you think, because the tumor can eventually grow, so the tumor must have some mechanism to prevent the attack of the lymphocytes if the lymphocytes is bad for tumors. So what we are going to do right now, or what the whole biomedical field in the world is going right now, is to find some compound or drugs that can improve the anti-tumor immunity, which I will introduce. Okay, so this is a very nice experiment done in 1984 by a very famous surgeon, Stephen Rosenberg, at the National Cancer Institute. That he transferred some immune cells into a mouse with melanoma. So this is the control. You can see that melanoma metastasized to the lung. But here it is the cell that the lymphocytes that are being adoptively transferred to the mouse, leading to the restriction of the growth of the tumor cells. This is a very nice experiment done in 1984. That's from that time we understand that lymphocytes actually can kill tumor cells. And I honor a very interesting figures, numbers from this figure, which you see. Seems not working very well. Let's see. Oh, okay. Okay. You can see this only a $1. So at that time, publishing a paper only cost $1 US. As comparing today, you know, we need 2,000 or 3,000 US dollar to publish a paper. So this is a huge difference. Strange. No. I know why. So that's a little bit here. Let me see. Okay, it works now. Okay, So as you can see here, even we have this powerful lymphocytes, but the tumor eventually can grow. So why? It's because very one important thing, which is discovered in the around 1990s, that these two very famous scientists, James Allison and Toshiko Honjo, they discovered very important, two very important molecules. One is CTLA4, the other one is PD-1. So these two molecules play a very important role to suppress the anti-tumor immunity. Okay. Okay. Sure. Okay. Okay, this is, you can see here, this is the tumor cells, and this is the lymphocytes. The lymphocytes have these two important molecules, PD-1 and CTLA4. So what are the roles? Actually, after our infections, so if we are infected with a virus, say COVID, so we have a lot of immune cells to target the virus or the virus infected cells. But the immune system cannot stay high all the time. If the immune system stays high all the time, the immune system will then attack us. So that's why we have a mechanism to turn the immune system from low to high and then back to low, the low level, which is by the PD-1 and then CTLA4. So which is we call immune checkpoint. So this immune protein is very important to act as a brick of our immune system, so that when our immune system was in a high level, by PD-1 and CTLA4, the immune system will go down. And then, so that, of course, it must be after the clearance of the virus or the virus infected cells. Our tumor cells are very clever. Our tumor cells have these two ligand, PD-L1, which is the ligand of PD-1, and then B7, which is the ligand of CTLA4. So the tumor cells use these two molecules to activate PD-1 and CTLA4, so that the tumor cell will give a break to this T cell and tell the T cell not to attack it. So the tumor cells are very clever. And because of this, of the discovery by James Allison, which is James Allison discovered CTLA4 and Toshiko Honjo discovered PD-1. So that right now we have two very important antibodies. So anti-PD-1 and an anti-CTLA4, which we call immune checkpoint blockade, to disrupt the interaction between the PD-L1 and PD-1 and then B7 and CTLA4, so that the T cell can reactivate and attack the tumor cell again. So this is a so important discovery. That's why in 2018 they received the Nobel Prize for the acknowledge their discovery of these two important antibodies. So right now we have several this kind of antibody approved by the FDA. I use bladder cancer as an example because I'm working on bladder cancer in my laboratory. So we have a lot of this kind of, for example, we have acilipsumab, which is for the treatment of advanced or metastatic bladder cancer. We have durvalumab, we have avelumab, we have several of these antibodies from different drug companies. Okay, let's see a very recent clinical trial published in the New England Journal of Medicine. So this is the best medical journal. Okay, this is a phase 3 clinical trial of avelumab to see what is the response. Okay, so this is a clinical trial called Javelin Bladder 100 trial and registered in the NCI website. So as compared to control, which is the standard treatment for the advanced or metastatic urothelial carcinoma or bladder cancer. So, and then this is the survival curve for the treatment treated with avelumab. You can see that the overall survival increased from 14.3 months to 21.4 months. And this is also the same for the patient with PD-L1 because this antibody targets. So for the antibodies to work, the patients, the tumor cell must have this protein or this protein. So if it doesn't have this protein, then the antibody cannot block the interaction. So you must have this protein. So when they look at, when this papers look at the PD-L1 positive patient, you can still see that the patient treated with avelumab has a much better survival than the control group. But not, there's one important thing is, it is not 100%. So not all the patients are being cured by this avelumab. So all the oncologists and the whole clinical world is now finding a solution. How to push this curve to up right here, because this is a very expensive treatment, very, very expensive treatment. But there's one issue that this treatment, as compared to the chemotherapy, is if the patients are being killed, it's being killed, the tumor will not recover again. So this is a very powerful treatment, but only work in around 50% of the patients, or even less. So the whole clinical world is to think why and how we can boost this treatment to a much better level. So to understand this, we have, sorry, maybe next slide, please. Regan, next slide. Okay, so to understand why, we have to understand a very important theory of how this immunotherapy works, which we call hot-cold tumor theory. So what is a hot tumor? What is a cold tumor? So for the immunotherapy, for the immune checkpoint blockade to work, so we must have a hot tumor. What is hot tumor? A hot tumor is a tumor that is inflamed, is inflammation. The tumor is able to release a lot of chemokine that attract the immune cell to infiltrate into this tumor microenvironment, which we call hot tumor. But for patients that doesn't work with the immune checkpoint blockade, they are in a cold tumor setting. The cold tumor means the tumor is not inflamed, it's not secreting the anti-inflammatory molecules to attract the immune cell. You can see there are more immune cells, there are less immune cells here. So for the immune checkpoint blockade to work, it must be in a hot tumor setting. So right now, the clinical world is thinking how to turn the patients that doesn't work with the immune checkpoint blockade, or you can say in a cold tumor, back into a hot tumor. If we have some compound that can turn cold tumor into hot tumor, then the immune checkpoint blockade will work. So what we are working at in our lab is to find some compound that can be an epigenetic inhibitor. So because people understand, this is the review that I written together with a scientist in the Netherlands, that we found that how to fire up a cold tumor is because our cell has a alter epigenetic machinery. So if we can change the epigenetic machinery, we can turn the cold tumor into hot tumor. Okay, so what is epigenetic machinery? So epigenetics is a heritable changes that can modulate our chromatin organization and gene expression without changes in DNA sequence. The ATCG sequence doesn't change, but the gene expression changes. So we have two very important epigenetic machinery. One is we call DNA methylation. The DNA are being methylated. Okay, remember this is a natural event. This is a natural event, but the cancer cell make use of this event to turn off some of the important genes that play a role in the inflammation or anti-inflammatory response. So the DNA are being methylated, and we have some DNA inhibitors, such as this one, 5-aza-deoxycytidine, which has a structure which is very similar to cytosine. So these chemicals may be incorporated into the DNA during the DNA replication process, and then affect the DNA methylation. This is one. The second thing is the histone modification. The histone can be modified. You know, the DNA are being wrapped by the histone molecule forming a nucleus. So these molecules can be this can be sorry for the interruption. So this is the process that we end acetylation. The removal of the acetylation, the chromatin will be compact and then affect the gene expressions. It's very important to or to inhibit the HDAC protein, which remove acetylation. So right now we have a lot of, for example, curcumin is one of the head inhibitors. So curcumin is in a lot in curry. Yeah. So if you eat a lot of curry, then probably it can help opening our chromatin. So but today, what I'm going to talk is mostly on the HDAC inhibitors. So we have some compounds that can affect the HDAC proteins so that the chromatin can be turned on again by the cancer cells. Sorry, can you help me to press? Yeah. Okay, so let me take an example of how those epigenetics inhibitors work to reactivate our tumor immunity. So this is a very nice paper published in 2015 Nature by a very famous scientist, Wei-Ping Zhao, in the University of Michigan. So what he did is he injects some mouse ovarian cancer cell into a wild type mice. This is a mice with the intact immune system, a normal immune system. And then he injects the drugs, the epigenetic drugs. As I mentioned, this is a 5-AZA-DC, and the others is another drugs that affect. This is a ECH2 inhibitors. So it affects the chromatin structures. So what you can see that this is the tumor size using the fluorescent signal. That combination of the treatment reduce the tumor dramatically. You can see here. But when these tumors are injected into the NSG mice, NSG stands for the mice without any immune system. There are no T cell, no B cell, no NK cells, nothing. So this is a immunocompromised mice. So when the tumors are injected in the NSG mice, you can see that the combination of the drugs had no effect on the tumor size, suggesting that the effect improve the immune system, but not the toxicity. Okay, actually, is this the case? The combination of the drug you can see it infiltrates. Why this infiltration of the cell can result? Because the combination of the compound increase two very important chemokines, which is called CXCL9 and CXCL10, which this is this chemokine that increase the infiltration of the T cells. Next slide, please. Okay, so the implication of this study is that our tumor is supposed to be able to release this chemokine, which attract T cell, and then the T cell can infiltrate into the tumor, so that when you use the immune checkpoint blockade, it can kill the tumor. But however, because of the epigenetic machinery, the CXCL9 and 10 are being manipulated and shut down, and it cannot release the chemokine. So that there are very few T cells being infiltrated, and then resulting that no response of this PD-L1 blockade. So this is very important that if we can make use of the epigenetic machinery, then we can restore the status of this epigenetic status of this chemokine from this state into this state, so that the chemokine can be re-expressed again, and then the T cell can infiltrate. Okay, so this is the example from T cells. But as I mentioned, our immune system has a lot of cells. We have T cell and we have NK cell, so which belongs to our innate immunity. So innate immunity means we don't need to teach them. For the T cell, we have to teach them. But NK cell, which belongs to the innate immunity, you don't need to teach them. NK cell recognize tumor cell readiness and kill them. So the NK cell, next slide, please. The NK cell is able to recognize the tumor cells by this the receptor NKG2D on the NK cell, as well as the ligand on the tumor cells. So this ligand is not expressed in the normal cell. So the NK cell will not kill your normal cell because normal cell doesn't have this. But tumor cell express this ULBP protein, something like that. So that the NK cell, once recognize this ligand, can kill the tumor cells. But tumors are so clever, so they shut something down. They shut it down, so that the tumor cells cannot be recognized by the NK cell, and then it cannot kill the NK cell. Even NK cell is there, cannot kill the tumor cells. So this very nice experiment shows that after treatment with the HDAC inhibitor, you can see by flow cytometry, the ULBP protein are being restored from here after treatment is to here. And there are several ULBP, and ULBP2 seems to play a much better role. That's why we want to see whether in bladder cancer we can see the same thing as well. Okay, so let me introduce some compound that we use in my experiment. So as I mentioned, we have a lot of HDAC protein. So HDAC 1, 2, 3 probably play very important roles in controlling the ULBP protein. So that's why we use HDAC inhibitors called entinostat, which can inhibit the HDAC 1, 2, 3. And we have another compound called cyproheptadine, which you can purchase, or your physician probably will prescribe these drugs to you if you have an allergy. So this is the very first generation antihistamine called cyproheptadine. It's a very simple structure. So our previous experiment shows that this cyproheptadine, or in short CPH, this CPH can have anti-tumor effect by inhibiting the epigenetic machinery. So we have recently published this paper that this very simple compound and very cheap compound that can restore our immune system. So in my experiment, I want to test the effect of these two compounds of turning on back the NK cell, the anti-tumor immunity of the NK cell. Okay, here, what is the experiment? That's we first did a RT-PCR experiment to examine the expression of ULBP protein, mRNA. So you can see that this is the normal cell. So and then you can see that those tumor cells didn't express the ULBP2 protein. It has a lower level of the ULBP protein, which is a very low level. Next slide, please. Okay, so here, after treatment with the atinostat, you can see the re-expression of this ULBP2 protein. And then in this is the mRNA, and this is the protein level. You can see upregulation. And then we also treat the cell with CPH, the the cyproheptadine, the anti-allergic agent. You can also appreciate that it has a upregulations of the ULBP2 mRNA as well as the protein level. So here we use this compound. We can demonstrate the upregulations of this ULBP protein in terms of mRNA and protein level by HDAC inhibitor as well as the CPH, this anti-allergic agent. Okay, so to demonstrate whether upregulation of this protein has any effect on the NK cells, so we culture the cells together. So probably you haven't done any cell culture here. So Sabrina, your PhD student in Professor Gunawan's lab, so she's in my lab right now in Taiwan. So she learned how to do cell culture now, and she knows how to do. She's doing the cell culture very well. Maybe in December when she comes back, she can teach you how to do cell culture. Okay, so we culture the tumor cells and then the NK cell together, and then to see whether treatment of the drugs can increase the effect of the killing of the cells by the NK cells. So we culture them together. But how do we know that the cells are being killed? So after treatment of the drugs, we incubate the cancer cell with this compound called calcium amide, which is fluorescence. So it will stay inside the cell. When the NK cell, if the NK cell is able to kill the tumor cell, this compound will be released into the medium, and then we can use this ELISA plate reader to see the signal released from the tumor cell. And we know the tumor cells are being killed by the NK cells. Okay, this is the experiment. We use different ratio of the NK cell to the tumor cells culture to do this co-culture. Okay, so this is the control experiment using the DMSO as control. So because the HDAC inhibitors is dissolved in DMSO, which I think you know this chemical very well in your experiment, probably you will use. So on increasing the ratio, what you can see that there's an increase in the cytotoxicity of this bladder cancer cell. But after treatment with the atinostat, we don't see a further increase. So we are curious why treatment of the extract inhibitor doesn't have increase of the killing. So we think one of the reasons is probably the NK cell may not express this NKG2D protein. Sometimes the NK cell express very low level of this protein. So we check, okay, yes, this is the case. The NK92 cell that we use doesn't have this NKG2D protein. Without this interaction cannot be set up. So that's why we did a very powerful assay called CRISPR assay. I don't know whether you heard of CRISPR, which received the Nobel Prize last year. That this is a genomic editing assay that you can mutate any genes, or you can knock in, knock out, you can manipulate your genome by using this powerful CRISPR assay. So we use this CRISPR assay to activate NKG2D. So we call this NKG2D knock-in NK92 cells. So now you can see that it has high level of NKG2D as compared to the parental NK92 cells. So we use these new cells to do this experiment again. Next slide, please. Okay, so this is the original data. And then this is using the new cell. So this is the DMSO experiment. You can still see an increase in the level on the cell ratio, on the NK cell to the tumor cell ratio. You can still increase, but you can see that actually the level is much higher, right? This is the original NK92 cell. You can kill tumor cells up to 40%. Up to 20-something%, but right now it can increase up to 40%. But importantly, after treatment of the cell, so we can see adinostat shows a much better killing as compared to the original one. Not only that, we can, when you treat the cell with CPH, the anti-allergic agent, so you can see that treatment with CPH also enhance the killing of the NK92, this new NK92 cells to UMDC3 cells. Okay, so to prove that this ULBB2 is actually responsible for this killing, so we use genetic engineering to overexpress, to put back the genes instead of using drugs. So you use genetic engineering, you use cloning to put the drugs, to put the genes back into the cell, so that the cells express high level of ULBB2 instead of using chemicals. So we use genetic engineering, molecular biology techniques. We overexpress the genes. So overexpression of the genes, you can still see enhancement of the killing. As compared to the cells without the overexpression of ULBB2, showing that this ULBB2 is actually very important for the killing of the NK cell and the tumor cells. And then those are for the human experiment. So right now we want to move into a more in vivo assay. So we perform an animal experiment. So, but for animal experiment, we have to use animal tumor cells. So we test this animal tumor cells, MB49. This MB49 cells increase ULBB2 expressions after treatment of CPH. So in animal, this is not called ULBB, it's called RNBT1, which is the hormone of ULBB2. So after proving this, that CPH works, so we inject the tumor cell into the mice, and then we treat the mice with CPH or the immune checkpoint blockade to see what happens. What you see is that, so the tumor size grows dramatically without any treatment. But you can see that after combination treatment, the tumor size decreased quite a lot. So when we check for the infiltration of the NK cell, you can see that the combination treatments actually improve the infiltration of the NK cells as well as the T cells. The T cells also increase so that the tumors are being turned from the cold tumor into a hot tumor. And then it can restore the whole anti-tumor immunity. Okay, next slide, please. Sorry. Okay, so this comes to the conclusion that epigenetic machinery plays an important role in tumor immune escape in both adaptive and innate immune response, which shows that it's important for the T cells, especially for my own experiment for the innate immune response, which is the NK cells. And then NKG2D ligand, which is ULBB2, is epigenetically silent in bladder cancer. And re-expression of ULBB2 by this HDAC inhibitor, this compound, can enhance NK-mediated cytotoxicity in vitro and in vivo. And lastly, so this epigenetic therapy able to enhance tumor immunotherapy. So what we propose right now is that if you have some compound, actually Sabrina is testing this. So we are using the compound that Professor Gunawan provided to see if the compound can increase the expressions of those chemokines, the expressions of the ULBB2 as I mentioned, so that the T cells, the NK cells, the inflammatory response can restore. And then we can have the treatment of the immune checkpoint blockade probably will respond better in those patients being treated with epigenetic inhibitors or some natural compound. Okay, so at the end, I would like to thank all my students, collaborators to do the experiment. Most of the experiments are performed by Himani and Nina. Himani is my PhD student from India, and Nina is a master student from Taiwan. And then we have some collaborators from the Academia Sinica in Taiwan, National Cheng University in Taiwan, and also University of Lausanne in Switzerland. Okay, thank you. Terima kasih. So I am happy to answer your question both online and offline. If you ask me questions, you have gift. Okay.
Okay, thank you very much, Professor, for the friendly topic and great speak. Okay, for the next time is the answer and question. We have some question, ladies and gentlemen, from the offline. We have maybe from offline two question, and the online is 2 questions. You have some question? Offline? Okay. Yeah. One for the next the left question. We have some question from the offline. We have maybe from offline two question, and the online is 2 question. You have some question? Okay. One in the right side and the left side have a question. Okay, again, you want to take a question? Okay, one in the right side and left side of the closing. Question. Okay, you can take question.
Good morning, Professor. My name is Leonardo from India. I am a student in the degree in chemical engineering ITS. So I have a question. So this compound is supposed to be enhancing the NK cell and T cell for detecting the tumor and killing it. So as Prof mentioned that in a system where the immune system is low or which is the T cell and NK cell cannot like be affected by the compound to have a direct effect to kill the cancer or tumor cell. But in first I want to ask, how is the effect on someone with maybe a history of autoimmune diseases, like they have aggressive immune system, how this compound can impact? Thank you.
You asked a very good question. So one of the drawbacks of this compound, as you mentioned, that so it increases the immune system. So it may have the possibility of turning on the autoimmune disease. You are exactly very right. So right now, they're undergoing clinical trial to use the optimum dose that will not trigger autoimmune disease, but will kill cancer. So the dose and the timing of using this compound is very important. So you're using very strong amount, so the immune system will turn high all the time, and it will be autoimmune disease. But on the balance, of course, you have to treat cancer first. So the main purpose is to treat cancer. And then if autoimmune disease really comes, then they have to treat autoimmune disease. But cancer is more than, so they have to treat cancer. That's why. But you asked a very, very important question. This is a very good question. Yeah. So let me give which one you want, a blue or red? The blue?
Red one.
Oh, red one. Okay. Okay.
Thank you very much.
Yeah. Let me take a photo.
Okay.
Very
Thank you very
good.
much, Professor. Okay, one question from the left side. You have a question?
Pak Hakun, Mr. Hakun.
Yes.
I from biology ITS.
Oh, from biology. This
Yes.
Dr. Awi is the
Yes.
19.
Yes.
Oh, yes. Hi.
You're welcome, Prof. Chan. Excuse me. On the another next. I remember. Prof. Chan visit in biology on 1918 nih, and my question is, and there are many compounds, yes, on the adapted and innate for immune response. Maybe there are characteristic, or maybe the for the adapted and innate, what's different? That's compound.
So you are asking how this compound are being developed?
Yes. Yes.
So most of the compounds that are developed based on the structure of the structure of the protein.
Okay.
So as I mentioned, the HDAC inhibitors. So they are all those compounds. You can see they are small molecules, which is hydrophobic.
Yes.
So they develop this small molecule based on the structure or the active site of the HDAC protein, so that the compound can block the activity of the histone acetylase activity. But for CPH, we actually don't know. So we're actually working on some experiment to identify what's the main target of CPH. HDAC inhibitor is very clear. It's affecting the inhibiting the HDAC activities, but CPH we are still finding the mechanism behind how CPH affect the epigenetic machinery.
Okay, thank you, Prof. Chung. Maybe the method in your research is by in vivo and in vitro. Maybe before in vitro we make a docking of the compound. It can do this docking compound docking molecular for compound.
On the docking. Okay, we didn't do the docking experiment.
Yes.
So because I am a biologist, so maybe I can collaborate with you if you are interested.
Yes.
That you can perform some molecular docking experiment.
Okay. Yes.
Yeah.
Thank you.
Yeah. Dr. Amin is the topic in your cancer.
Okay. Yes.
We have like chemical engineering lecture in online, Professor. Yeah, he studied in the Australia. Now is Mr. Doni. Do you have some question, Mr. Doni? Please.
Hello, Professor Chan. Thank you very much for the wonderful presentation. That's really interesting. It's an honor to be able to attend the seminar that you had this morning. I just have a quick question regarding the presentation. You have successfully tested the effectivity of the compound to the animal, right? What do you think would be the percentage of, the successful percentage of the compound when it comes to, being applied to the human in the near future? Thank you.
Yes, this is also a very important question. That's the compounds are now being tested in the clinical trial. So I have a review papers that's it's been successfully tested on breast cancer patients, that it can increase the efficacy of immune checkpoint blockade in breast cancer patients. And we are still the clinical setting is still under a lot of clinical trial to examine the activities or efficacy of the combination treatments of the epigenetic inhibitors plus the immune checkpoint blockade. So some of them are still in the clinical trial. And actually, if you follow those this recent AACR, last year AACR meeting, that's a lot of researchers are working on how epigenetics inhibitors affect the lipid metabolism. The lipid metabolism plays a very important role in the anti-tumor immune response. So if it can be blocked by the epigenetic inhibitors, so the anti-tumor immune response using immune checkpoint blockade can be increased. So, but in summary, a lot of those epigenetic drugs are still being tested clinically. And then I don't have a definite answer of what is the future, but I am myself very promising on those epigenetic drugs.
Thank you very much. That's a very important insight that I can learn from that. We can probably collaborate in the near future, because I myself I'm working on the biomedical applications of bone implants and those kind of things. So that's probably going to be such an interesting pathway to look at in the future. Thank you very much, Professor Chan, once again, and thank you, Pak Hakun, for the time.
Thank you. I'm really happy to collaborate with you.
Yeah, chemical engineering senior professor.
Jam 1 lah, hari biasa ini loh.
Oke.
Enggak lah.
Oke.
Good morning, Professor Chung. I'm from the Department, IPS. So my question is about your anti-cancer compounds is, have you ever tried or practiced the antibody drug conjugates? Because you tried it in Afilizumab. It's targeting like a very specific target for the tumor. Have you ever tried that? And maybe we can like give a compound to the antibody drug conjugates. Thank you.
Thank you. Your students are really good. They asked a very good question. So antibody conjugates are very important technology right now. But for me, I haven't tested, but in bladder cancer, we have several antibody conjugate right now that are being used. So it's FDA approved, being used. But for me, I'm not doing that. In the biochemical engineering department, I have a talk on Wednesday. I have a different talk, which will, but it is not antibody conjugate, but this conjugation between some DNA and then a gold nanoparticle for the detection of cancer. But something similar, but not exactly the same of what you are asking. But ADC are very important right now. So the avidumab, as you have mentioned, actually tagged to several drugs as well in the clinical setting. So you asked a very good question. You want a red or you want a blue? The blue one. Okay, I have red.
Thank you very much. Thank you very much. Victor. Okay.
Very good question.
Maybe in the next time you can take the PhD and master degree from IPS, Professor.
Sorry, can you say that again?
Maybe in the next time you can take PhD on master degree from IPS.
Yeah, I would like. Yeah.
I didn't see from Indonesia.
Yes.
Okay.
So I still have some.
You,
Okay, you want to ask questions?
have a question again? Again. Okay.
Rick has question.
You can have question.
Check. Okay. So good morning, Professor Cheng. Good morning to Mr. Hakun and ladies and gentlemen here. So I would like to ask some questions regarding to the presentation of Prof. Cheng today. So my first question is it possible for us to get the anti-cancer compounds from natural sources such as foods and vegetables? If so, how we polymerize it or how we change it to become an anti-cancer compound? The second one is it possible for the anti-cancer compounds to have side effects in our body, especially in our liver and our kidney, if we use it, let's say, more than the recommended dose? So that's all from me. Thank you very much.
Yeah, this is also a very good question. That's actually there are lots of natural compounds have anti-cancer effect, but as far as I know, that most of the compounds work on the anti-angiogenesis. So as I mentioned on the very first two slides, that in the tumor cell we have blood vessels. So a lot of natural compounds affect the growth of the blood vessel. And of course, some compound itself has anti-tumor activity. But as far as I know, there are a lot of compounds working on anti-angiogenesis. So for the compound working on the immune system, I'm not very sure. But I remember last time when I come here to share, I share some traditional Chinese medicine, the TCM, which can increase the immunity. So last time I shared, not exactly the same pathway, but it increased the immunity, it affect the inflammation. So regarding the dose, I think this is also important that if you are using too high level, it still have the toxicity. If you purify the compound and then take it, so that's why we have to do testing both in vitro on cell line and in vivo on the animal to test the toxicity to the cells so that we won't overdose. So for immunity, it's very important is the balance. The balance. So if you boost the immunity too high, as I mentioned from your colleagues, that it may trigger the autoimmunity. So that's why we have to do the balance. That's important. Very good. So you want a blue or you want a red? I guess blue is one blue. I thought we would say he won 4. So is it red, right? Okay.
Thank you very much for the
Okay. Good.
okay. Okay. Some question on online or the offline?
For fellow faculties, I will give you this blue,
Yeah.
Dr.
Okay.
Hakun.
Maybe Dr. Abi, we can go to biomedical on Tuesday at 1 p.m. Is it okay?
Oh, okay. We want to go to Teknik Kimia.
No,
Engineering.
on Thursday.
On Thursday. Thursday.
Biology department on 1 p.m. Is it okay?
Yeah, okay.
To take
Okay.
Soto Chahan.
Oh, yes, yes.
Okay, okay, thank you very much. Do you have some question?
One last.
One last.
One last. No more. I have to say to others.
Maybe after that, from me, Professor.
Yes, I say. Yeah, I have to say some for Mr. Hakun. So this is the last one.
Thank you very much, Professor. You have some question? I have some questions, Professor.
Please.
It's the experience for my mother. Because my mother have cancer in the colon and take operation in 3 years ago. Is it that the cancer virus can develop again in the human body? And what is the compound that can increase the activity of the cancer?
Yeah. So you mean compound that increase the anti-tumor, right? So for colon cancer, right? So for colon cancer, as far as I remember, so they use the first line is the 5-FU related chemotherapy right now, and then it has to check, if I remember correctly, that the tumor cell has to express for targeted therapy. For targeted therapy, the tumor cell has to express the HER2 protein, which is the EGFR receptor. So if it has the EGFR receptor, then we can use Herceptin or related small molecules that can block the signal. Yeah, but I think I would suggest maybe you can go for some diet control and some natural compound that can boost our immunity. Yeah. But sorry to hear that.
Because what the stress is can increase the activity brother. Stress.
Stress. Oh, yeah, stress can increase.
Okay, thank you very much for the sir. We have some question, people, audience. Offline and online. We have some question. The MC have okay, one. Okay.
Excuse me, I have a question, Mr. Chung. So the compound that you make or that you discovered, it can increase the anti-cancer immunity, right? My question is, let's say, so breast cancer only happens in women, if I'm not mistaken. How young does the person need to be to take the compound? Is that ever considered? Maybe like if it's too young, maybe it will cause some detriment to their growth, or also it increases cancer immunity, right? So that means if they already are afflicted with breast cancer, does that mean, well, that's it. Yeah, that's all my question. Thank you very much.
Yes. So I think you are asking that how early the patient has to take the compound, right? So I would suggest you have to follow the standard chemotherapies on the very beginning. So after the standard chemotherapy on the adjuvant treatment, I would suggest you can go for the immunotherapy, and then plus or minus with the epigenetic therapy. So, but for the first line treatment, I would suggest going through the standard clinical treatment. Yeah. So I cannot say how early it is. It depends on different patients, different staging. So it's completely different. So I would suggest discuss with the clinician, discuss with the oncologist. To find the most optimal treatment for the patient. Yeah. So this is the last one, okay? This is for you. Hey, one more. Oh. Very good.
Okay, thank you very much. For the online, have some question. One question for the online. Oh, the offline. Okay. Okay. What's your still please? So the souvenir from me, Professor.
Okay, yeah, sure. Sorry, I have to say the souvenir to Mr. Kun.
Yes.
So you don't have souvenir. I appreciate your question.
Thank you.
I will ask, why you choose breast cancer other compared with other cancer? Why you choose breast cancer then compared with other cancer? Like means breast cancer. There's a lot of cancer when you choose a bladder cancer.
Why I just work on bladder cancer? Okay, so because bladder cancer is an important cancer in my city, so a lot of patients have bladder cancer in Jiayi. So that's why I work with the urologist to find some compounds and treatment for bladder cancer. So why bladder cancer has a lot of, why Jiayi has a lot of cases of bladder cancer is because of the drinking water. So drinking waters have arsenate. The arsenate, yeah, arsenate, AS, yeah, AS. So in Jiayi, so this arsenate is known to induce bladder cancer. So not a regular drinking water, but the water from the well. underground water. Some of the residents drink underground water, which is contaminated with arsenic. So the arsenic is come from the mineral soils, but not pollution. But in Chiayi area, we don't know why. I have to ask the environmental earth and environmental science or geologists. We don't know why, but Chiayi has quite a contamination of arsenic in the underground drinking water. That's why we have such a high case of bladder cancer in Chiayi, but not in other districts in Taiwan. So that's why I work on bladder cancer, but I work on some other cancer as well, because I have some students, they are oncologists. So they work on oral cancer. So in Taiwan, there are lots of people chewing the betel nuts, the betel nuts. So they have oral cancer. So I have patients, I have clinicians, they are my students, they work on oral cancer as well. But good question.
Yeah. Thank you very much, Rosa. For the online, have some question? Enough? For the online? It's a good topic. Because Indonesia now is for the cancer is the 5 top disease in Indonesia. Usually more than money to cover the cancer. So this one, so it's a very important for us to take the cancer disease development. Okay, you have some question? Okay. No? I think the question. No? Okay. Okay, I think if you don't have a question, so it's a very, very good topic. And know that the compound that can decrease the cancer and what is activity that can increase the tumor activity. I think it's a must important for us here. Indonesia, as I mentioned in the previous, that have too many people take cancer in the body. I think must know about how to handle this cancer. I think this is enough for our presentation. Could applause for the Professor Chung. Thank you very much, and I take to the slides of ceremony.
Thank you very much, Prof. Chan, for the information. It's really an honor for us for having you here. Okay, before I close the event, I would like to remind you that we also have the second session of this World Class Professor 2022 will be held tomorrow at 1:30 p.m. The topic will be about how to write manuscript and answer the reviewer. I work as a college student. It's very useful for us. So I hope you guys can make some time tomorrow. Ladies and gentlemen, thank you for your kindly attention. Maybe we can take a photo together with Professor. So the audience be and please come up front. The audience can please come in front of to take a photo together.
So where should I stand? Stand here.
Okay.
Maybe the girl can be at the front and the boy at the back, please.
You mean university? Okay, okay.
Okay, here. 1, 2, 3. Okay, one more. 1, 2, 3.
Excuse me, about our university and our program that you are free to take. And then we have full scholarship for our College of Science. So, yes. Some people take very low dose every day to prevent but I think we can take someone that's like
Isang hindi pa ako ngayon. Okay.
so, as I mentioned, under the knowledge,